So welcome you all for the session. One of the hot topic is analytical techniques and drug interaction, which is one of the prerequisite for integrative medicine, development of integrative medicine. So today's chair is Professor Y.K. Gupta for the session, and I'm very glad to introduce Sir. Sir is a professor and head in AIMS, New Delhi. Sir has chaired many organizations. Sir is president for Indian Pharmacological Society. Sir is involved in designing Schedule Y as well. Sir is advisors to the various WHO agencies focusing natural product drug discovery as well as modern drugs. Sir was dean to the Indian Society for Rational Pharmacotherapeutics and many more to name few here. So please welcome sir. So this session is focusing on first part is analytical techniques. So analytical techniques has huge potential to standardize the herbal drugs, complementary alternative formulations, which is the fundamental and first most step required in drug discovery and development. So cut it short due to the lack of the time, I would like to invite first speaker of the session is Dr. Lal Hingorani sir. Welcome you sir. Dr. Lal Hingorani sir is a director and chairman for Herbal Pharmanza Private Limited. Sir has done his PhD from IIT, and sir is not only organic chemist but he's a phytochemist and working in the field of herbal pharmaceuticals. And sir has instrumental in understanding regulatory and industrial perspective. So I welcome you sir for the talk. Thank you. Please, sir.
Mainly listening at last 2 days, all these hot topics from Ayurveda and all these things, I was a little worried because I probably do not know A of Ayurveda much. I just go for these Ayurvedic sessions, pick up their mind, pick up what is there in probably what they were talking about, taking away the thing from them. We develop the products for the Western market, and our company has started basically for just Western markets only. About 95% of our turnover is exports of the bulk extracts, which are definitely standardized. They have a lot of clinicals, and a lot of work has been done. And each product, about 10 of our products are doing quite well in the international market. Few of them I'll show you also. And I talk about the opportunities that companies have, like, and it will be on the companies how they can do a business in India as well as abroad. Natural products for health. There's a history. You must have heard a lot of it. Egyptian, Indian, Chinese, all these things are there. Then para-herbalism, what you called sometimes even Jhola Chap, those people who are sitting on the streets selling certain herbs as such, or even animal products for that. That is, then there's a traditional system which came, that one of this is Ayurveda. Then philosophy came that the natural products which are made into the plants are because those plants want some compounds to be synthesized which are good for them. And that's what we are picking up for our own use. And use of herbal medicines by animals. There were people who were observing animals, what kind of herbs they eat when they are having certain kind of problems. Even they have studied the birds. When they form a nest, they'll put certain kind of herbs in the nest, so it is having antibacterial properties. So when their offspring comes up, it doesn't have any problems in that. So these things are like a very well documented, and they have observed that though there is no knowledge in them, but they know what to pick up and what to do, like for the health issues. Natural product drug discovery can be what it is meant for. That is a target. like and then is what for you are going to do this work, and second is regulatory requirement of the country where you are want to market that. Regulatory requirements of each country differ. Nowadays harmonization is occurring, but as yet harmonization has not come into effect. Then India, there are different classes: traditional medicines, what you call Ayurveda, Siddha, homeopathy, and other class of folk medicines. Nutraceuticals is another one which came up under FSSAI after a lot of going to Supreme Courts and all these things. Finally, it is set. They have come out with 400 herbs which you can sell very easily in Indian market. There are negative things, there are positive things. Negative things are they are not controlling the advertising of these herbs, and people are selling these herbs as Ayurvedic herbs under FSSAI and claiming a lot of things which a nutraceutical should not claim. So you see every day, full day, the TV advertisement is going on about these products. So this is to be controlled. And phytochemicals or pure compounds, like the ones Taxol and all these things, what has been Vincristin, Vinblastin, which has come out and formed a drug finally. So they are like registered as drugs only. And fourth one that has come up is phytopharmaceuticals. This will be a very good segment because this you can say Ayurvedic single hub, having a modern complete research done on it. If you pick up, now it depends on how you pick up a phytopharmaceuticals. You can pick up a product which is being manufactured as per the Ayurveda, find out the 4 components in that, what is their must. You find out 4 components in that, identify them, and then do the work again for a drug. Though it is, the restrictions which are there are very high, and I have just found out from the group which is working in the government of India that almost for one product to come out, it will require something like 6 crore of rupees of research grant. So either government gives, or small companies will not be able to do this, or big companies will be going into this. So a lot of products government has already taken it. We have also taken 2 of the products, one with CDRI, one with IIIM, and we are doing that for the phytopharmaceutical. But it will take 3 to 5 years still. And one of the biggest market of worldwide for the natural products is USA, and nutraceuticals and phytochemicals, these are the 2 only things. Nutraceuticals in that they have different segments. That is, they have some nutraceuticals where claims are allowed. They ask you for at least 3 good clinical trials and they give you a claim on nutraceuticals. If you do not have any good clinical trials, you can just sell it. For arthritis, you can say for joint health. You cannot say arthritis, so you cannot use any medical term in that, but you can sell those products for the particular, people will understand what it is for, like that. So that is one of the biggest markets from there. Now these ones, actually yesterday somebody showed this one, some of the diseases, health problems in India. Now it is in India also. Initially it was mostly the diseases by the bacteria or some infection. Those infection diseases are going low and low, and lifestyle diseases are coming more and more, which was there in the West. Now it is happening in India also. So anybody who wants to do research, they have to pick up this, what will be the market size, where they are going. Finally, when you are doing a business, you have to see that how much will be the market. And this is for the US. US biggest now is coming out to be Alzheimer's, and that has become a bigger problem. In India it was not there, and somehow somebody picked up what Indians eat regularly, that they are not getting Alzheimer's disease. It is various factors, because here the families and all these things are together, so emotional binding and all these things are there, whereas they just picked up, probably they take yellow curry every day. Everybody takes yellow curry, and that curcumin came up. Curcumin came up also for one more reason, that government of India had approached US to pick up some herbal products from India and do some research in NIH, and they had picked up curcumin for anti-inflammatory, but it went and ended up in Alzheimer's. So Alzheimer's, that is one of the biggest problems, and today this is the first product, curcumin, which is entered the world as #1 product. Last year it was number 1 product. This year it became number 2 because suddenly CBD came up. CBD has become number 1. 346% growth, which is our bhang. Now, I have specifically written this research prospects for Indian non-Ayurvedic companies for Indian market, because Ayurvedic companies, they do have their own products, they have their own systems, they go to the Ayurvedic doctors and give their products. But the people who want to develop Indian Ayurvedic herbal products, for them the research guidelines like NA will be little different and how to do it. Now, Ayush mode may limited to certain diseases where this is again debatable. Ayush people will probably not agree also. No medicines available in allopathy system. Give you an example of this. I had seen one paper from the CSR laboratories where they had done fracture healing property of Harjod, the Cissus quadrangularis, and it was something like 15 years old paper in which they had scanned 4 herbs. One was Cissus quadrangularis, second was ashwagandha, third was tulsi, and fourth was shatavari. And it was in one of the proceedings of CSR laboratory. It was lying. I just picked up that book from a library. I read it, and I said, this is Cissus quadrangularis is really interesting, because for fracture there is nothing available in allopathy system. And a person who gets a fracture, you have to sit at home. If it is for 2 months, a laborer will have that much of money gone, like, and they'll be without pay salary. So I said, this is the one thing. And nowadays, the old age may, now you see a lot of fractures and they become like immobile, and all the family suffers with that. So picked up that, and we did a clinical trial. And after the clinical trial, it was lying, nobody was coming up. Suddenly one orthopedic doctor came. He said, I want this product, gave him 50,000 capsules. He shut down his own practice and his hospital, and he went for this product. Single product, he launched a company, and it became 12 crore product in 2 years time. And then everybody came in. Today it is 100 crore product in India. Nearly companies like Alembic are selling it in this country. So we came out of that, we went for another product. So something like this, and this is, I'm talking purely business as well as research together. So this kind of something where nothing is available in allopathy systems, if that is picked up from Ayurveda, it becomes hit quickly. And then there are Supreme Court judgment, nobody has challenged that, so there is little restriction. If you go to the allopathic doctor, he'll say, but this product, Cissus quadrangularis, in Bombay alone, 800 orthopedic doctors are prescribing that, because nowadays they are not bothered, but because there is no alternate available, so they are taking it. So rather than writing that RX on that, they suggest you take this. So, but nobody has gone for this Supreme Court judgment, and then they say, and allopathic doctors have, because how we started is we went to the doctor, you have certain cases where there is nothing happening, try this out. We just gave them samples. And when they got results, they accepted it. Then there were a lot of this thing about quality and efficacy and all these things. Efficacy though they proved, but about quality and all these things they didn't care. But then we have a lot of certifications from US and all these things. We showed them all that thing. It became easy. But same thing is not there with the Ayush companies. Ayush companies as yet, because I was attending a conference of USP and Ayush in Delhi, in which one person was very bold and he said, let us increase the quality aspect of the Ayurvedic companies. And if the companies who are not able to come up to that standards, let them close down, let them go and do some other business, let them go and make bricks. That's what he said. Probably they'll get more money in that, because here you are playing with the life. Because there was a paper from Katyal, Dr. Katyal, for these heavy metals, like, and we immediately, whenever somebody from the US and this thing says Indian products have heavy metals and all these things, we react. But Dr. Katyal had picked up something like 12 samples from the market, and he had done this heavy metal, and that paper was published almost 15 years back. And in that paper, he has shown that how much heavy metals are there. Like, and when Indian government also took up after the thing came in Nature, they found out that nobody is washing the herbs, because here, like it's not that old time when the forests were ekdam completely without, they were bound by those people only, tribals only going there. Now everybody is going there, cars are going there. So a lot of pollution is there in that. So this lead and all this thing is coming, and because commercialization has occurred, now I'll give you an example of a ginger, sunt, everybody must be using. How do they dry it? Sunt, they just put it in your calcium oxide. As soon as the ginger comes, they put it in calcium oxide to dry it quickly, because it picks up moisture quickly. Then they dry it in the sun. So sunt we have analyzed, and it has something like 200 ppm of arsenic in that. That much, lead and arsenic both. So nobody sees here, because the commercial side is not educated. They do not know these things, and they want quick money. So they are doing what is fast, like. So that kind of thing. So if till the companies which are manufacturing do not analyze it, you'll be ending up getting these kind of products with us. Now, the nutraceutical mode market is growing, life expectancy is growing, and educated population is increasing, and they want good health. Nowadays, if you see all the people, they are looking for good health. And if you see quick, one of the products that I'll tell you, IIM-9, which was anti-diabetic, came up from CMAP. It became 90 crore product overnight. And he started advertising, take it with your allopathic product, whatever you are taking, and take this 5 rupees a tablet. It became so popular. So people want it, that's what I'm saying. And what happened to that product? It did not have any clinical trials, anything. It went down. But acceptance by people is there. If you have a very good product, that acceptance will come. So this is a very big market, and ease of marketing, law enforcement is less in this as yet. Phytopharmaceutical, very new segment, and we don't know what will be the market, because they are saying that allopathic people will be able to prescribe it and all those things, everything is fine. But what will be market size? Because it will be confined to only India. You cannot sell this product in a world market, because as yet world market, they are not accepted this phytopharmaceuticals. Till they accept this, you have to confine to this, so market is limited, like, for this. So we have to see. And then nowadays this Biodiversity Act has come, which is behind you. They don't want you to, they think the same thing, that you are taking away the knowledge of our this thing old system and exploiting that, and you should come to us. There are no guidelines very clear in that. We are fighting for that. Okay, how much or what? We say, you say 0.5% should be given for this. They are not saying that. You come to us, you come and sit with us, then you do an agreement with us, and they will decide how much you should pay. So this is this thing, we are fighting it out. The industry is fighting out this one. This one, international nutraceutical market is $250 billion for US in nutraceuticals. In that, these our spices, herbs, and all these things will be one of them. Like, the segment is growing. This is the total one, and 52% is US and Japan alone. So if you just concentrate there, it will be very good market for that. Indian export is only for this kind of segment is only $1.5 billion, which is nothing. So China is almost 10 times than this. So we have to have certain laws or this thing where we allow people to do research into this segment and go for that. Our strengths are our traditional knowledge, a lot of spices, herbs, tropical vegetables and fruits, like you have seen the success of curcumin, then success of piperine is also there. And now ashwagandha is in the first 10 herbs in the world. Ashwagandha is picking up. Initially only ginseng was known. People were talking about Indian ginseng and Indian ginseng, but it is not a ginseng. It's a different totally variety. Botanically it is totally different, and it is much superior than ginseng for the properties that they have. So infrastructure is there. We have a very good pharmaceutical company, so Our knowledge is there for making the nutraceuticals, and bulk extraction capacity is also there very much. How to go about it? AYUSH mode. If we have to go to the world with the AYUSH mode, it has to be government to government, because teaching them Ayurveda, like today I'm telling you myself, I have not learned in the last 20 years, probably 0.001%, though I sit very regularly with Dr. Ashok Vaidya and Dr. Bhushan Patwardhan and all these things. I've been sitting with them, talking to them, but I pick up only what I want. Like when they talk, I just pick up those things, what is relevant to the market. And I'm not here at this age to learn, because Ayurveda, you require probably two of your 200 years, 300 years, that's what I've heard, that much you require for the learning that. So AYUSH mode will be difficult. Herbal and nutraceutical mode will be much easier to go about for the export market, and traditional base we should take it up, and from there we should go do some work of making product acceptable to that country and doing some clinical trials. It is not that only single herb products are going into the world market. If you have a combinations also, 2 to 3 herbs combinations they are accepting, because they have little restriction, because they say if it is more than 3 or something, the analysis becomes much difficult in that to find out, and there the analysis is compulsory. You are saying that these 3, like we were selling 3 tulsis, that Vana and this Sham and Ram. These 3 tulsis together as a tea cut. They said, you just tell us that you are putting 1/3 of this, 1/3 of this, 1/3 of this. So finding out their total profile and giving them which is not there in this, which is not there in this, and on that basis we convince them that it is all 3. So this kind of work is required in that. Easier way in this nutraceutical market is bulk extracts. Now I was with Asochem, that's one of the IT sector ka industrial association, and they were saying when this thing came up, India wants to enter this IT thing about 20 years back. So they said, we were looking for what we should do. People said we should start developing the finished product, like Microsoft and all these things. So the industry said, we do not want that, because we are as yet not competent enough, and we'll do mainly is just give them support, and let that Microsoft and all these things do the job, we'll give them support. And today that is $190 billion industry. So that is a success rate. So in this also, if you see the gap which is there between the world market and what we are exporting, it's quite big. Even if we do little efforts, we can achieve a lot of good business in that. And then as per the regulatory requirement of the country, you have to go. Basic requirements of a natural product to enter any market is identity testing. Identity is becoming must. Recently, you must have heard that one of the, which was a wrong approach again, governor of the New York, he picked up certain herbal products and then he sent it for DNA testing. So herbal extracts will not have DNA. He was not knowing that. Herbs have it. So now identity is become must, and they have gone up to the DNA testing of that. Here we have a lot of, in Ayurveda, there are a lot of other things which we can do, and we are doing that, cross sections and all these things, botanists, they find out. And in India, government of India has done, which is not documented. They have done a test of what herbs Indian big companies are taking, and they have found out that 65% of the herbs what they are taking is not actually what they are writing on their label. They have done a DNA testing of that. So here, like so many people, small companies, they do not know even. Today, like I had ordered a Brahmi, like central acetic cake. So we know. So we will catch it, but there are companies who don't catch it. Then contamination, impurity profile, actives if known, pharmacology action, toxicity profile, stability of the product, and availability in sufficient quantity. If you are going to the world market, they'll ask you, today they might be buying 5 tons of that. They'll say, can you make 500 tons? They'll ask you whether that much raw material is there, because if the product clicks, they don't want to lose the market. And that much volumes are there. Like Brahmi Bacopa Monieri, when we started, there was a party which came and he said, I would like to buy 100 tons, do you have it? I went and found out how much is the raw material available, and mainly it is coming from Calcutta. It was like 100 tons of the raw material is available only in Calcutta. So those things we do not know, but they are there. So that kind of work has to be done before to go into these kind of markets. Synthetic dyes and all these things, they are mixing up metabolism, absorption in the body, and anti-inflammatory drugs which are being added into the curcumin. So a lot of negative things are coming up. I think I have less time. I'll just go through in 5 minutes. 2 minutes, okay. So I'll give you just one example of the curcumin. Curcumin Research Long Vida is our patented product, which we have licensed out from UCLA. UCLA had done a formulation of this for Alzheimer's. They showed that it is absorbing in the brain, and it is again, these are the different markers for the Alzheimer's, and they tested it and they said, it is working in most of them, because it was curcuminoids, 3 of the curcumins. Then the challenges of the curcumin was Cmax is not there, so they had some SLP formulation which is absorbing into the blood, and they showed that rather than going through the blood, it is going through the lymphatic system, it is getting absorbed. And we also did some work here, how it is going through this, and we even saw that PK study was done. We done small clinical trial on cancer also on this at Tata Cancer, humans. So that time this PK study was done. And Brahmi is another one which has a lot of properties in the brain segment similar to this. And for that we have done 2 clinical trials, one on Alzheimer's patients, one on the second year MBBS students. And we go with all these actives like these all bacosides. We have even tested them for their activity in the brain, and we found out which ones are the ones which are having maximum activity, and we see to it in our extract, they are the maximum. It's not single this thing, but it is multiple things are there. This is the one that activity we showed on the HT1A receptors. And then ashwagandha again, there are almost 10 methanolides we have standardized with and validated the method for analysis for that. This is the one. And this is again Vitaferin A is showing very good activity in cancer, so we have done some work in cancer. And both the products, curcumin and Vidania, they are in certain clinical trials in India, and they are also in USA. USA mein curcumin is going for phase 2 now for head and neck, and ashwagandha is going for phase 1 in the brain cancer. So it is not only Vitaferin A, it's a full in which the concentration of Vitaferin A has been fixed like that, and we have done all that toxicity studies and all these things on that. Thank you very much. These are some of the members of our and we are thankful to a lot of the people from the Ayurvedic fraternity, like from last 5 to 7 years I've been working with Dr. Ashok Vaidya, with Bhushan Patwardhan almost 8 to 10 years. So it's knowledge which is coming up, and we are just refining it and taking it to the world. Thank you.
So I request YK Gupta sir to felicitate Dr. Lal Hingorani sir. Thank you sir. I request Mr. Sharukh Bharucha, a senior advisor to Ancrom Private Limited, Mumbai. Please sir. Dr. Sharukh Bharucha is a basically organic chemist and consultant to the Ancrom laboratories. He is expert in chromatography, TLC and HPTLC, and being an advisor to the Ancrom, I request Dr. Bharucha to, in brief, explain the chromatography and TLC. Please, sir.
No problem. Good afternoon, everyone. As rightly said, I am not from organic chemistry, I am from analytical chemistry, and I have completed almost 40 years in pharmaceutical industry as a quality control person, worked in quality control, quality assurance and regulatory affairs. In pharmaceutical industry, the suitable tool for analytical work is HPLC. HPLC is a tool used for all chromatography work, that is for impurity profiling, stability studies, and even for quantification. But I have found, after retiring from pharma industry, this planar chromatography, that is thin layer chromatography, with modern instruments in place, is a very suitable tool for herbal analysis. For just now in the plenary session, there was a question, do we have researchers of good quality? Yesterday, with my presentation in Bharatiya Vidyapit College on planar chromatography, many researchers approached us for good work as far as research using planar chromatography as a research tool. Even FSSAI, they are also working in collaboration with us for developing proper profiles of herbs used in nutraceuticals and in Ayurvedic medicines. Before I start with the brief overview of this, I'll not say the best technique, but a very suitable technique for herbal work and material of botanical origin, I wish to highlight USP has included HPTNC in the USP 2017 for material of botanical origin by including Article 202, which is for oils, 203 for material of botanical origin, 2051 for material of contamination in natural products, and 1064 to do proper TNC work. What is the difference between HPTNC and TNC? TNC we were doing also in pharma industry, but TNC was used only for identification test, because at that point of time we never had proper instruments to do quantitative TNC. We never had proper SOPs. We never had proper instruments. Camac, which is a Swiss company, I found this to be very fascinating, brought about proper instruments to do proper analytical work of material of matrices which are complex in nature. API matrices are very simple in nature. They have only one or two main APIs which can be very well analyzed by using HPLC. But HPLC has its own limitations. In a brief presentation for next 10-15 minutes, I will show you how this tool can be used for herbal preparations, even for quantification of your botanical marker compounds which are found in your herbal preparations. This second slide, it was referred about ancient ginseng. See the difference. This is the difference between your conventional TNC and instrumental TNC. You can see how the metabolites which are present have separated out properly, have resolved properly, giving you the proper insight about the active ingredients which are present in your compound. Here, with proper application and band application, you can see the perfect resolution of your metabolites. This is the most important takeoff point for good analytical work as far as your herbal work is concerned, as far as your material of botanical origin is concerned. HPTNC and TNC. In TNC, we were only doing qualitative work. In HPTNC, we are doing quantitative work. I am telling you, if you have your botanical markers, you can even quantify them. You can even validate your methods. Since it is now official in USP validation of your methods, especially the people who are in the market of preparing medicines of botanical origin, and those who wish to go for exports, they have to have proper validation of their methods for their regulators. For proper validation, the most important test is the reproducibility of your method, the linearity of your method, the recovery of your method. All this can be well defined and well carried out by using proper instruments which are available. Initially, what we were doing in TNC was only the applicator, the development chambers, and the UV cabinets. In the UV cabinets, we used to just see the profile in UV lamp, fluorescent lamp, and in white light. And we used to capture only the images of the profile. This was at the initial stage. See how it has developed further. The instrumentation part of it now. Now, the applicators which were manual, which were not reproducible as far as the volume of your sample which you were depositing on your planar surface for quantification. They have been replaced by proper applicators in which you can do proper band application, maintain proper distances between the tracks which you are applying. You can apply as many as 15 samples on one planar surface and see the entire profile. This was absolutely essential for the manufacturers of herbal products, because even the regulators, they wanted on the same planar surface the material which has already gone to the regulated market, and what you want to put in the regulated market should be on the same planar surface on different tracks, developed under the same mobile phase up to the same distance, and then take the images, capture the images before derivatization, after derivatization, and send to the regulators. If you read the USP chapter 203, they have not said about quantification. They have said about fingerprinting. What is fingerprinting? Fingerprinting is nothing but the proper profile of your product. If the fingerprinting profile is accepted by your regulators, this is what FSSAI is also looking for. They are making proper fingerprints of all the authentic herbal samples which are available in the market, so that if the fingerprint differs, they can even find out if there is any contaminant added into the product. This became very important for the second instrument which you see, that is called the photo documentation system. No longer you can take the camera, take the images with your camera, but now there are proper photo documentation system to capture the images at different levels. Then came the scanner, very important, which is the heart of your planar chromatography. With the proper scan, you can do the quantification of your marker compounds of interest. Even the other compounds which you find in your profile, after doing the UV scan, you can take the UV spectra of those compounds. You can find out the lambda max values, and even after going to the library, you can identify those. There is one more stage available to do the better confirmatory state, which I will show you in my next slide. This slide shows you that initially all your instruments were working on stand-alone mode. These are now driven by a software which is absolutely necessary for any analytical work, whether it can be herbal, whether it can be nutraceutical, or whether it can be pharmaceutical. All your instrumentation work should be with proper software which are 21 CFR compliant. So these instruments are all working with a proper software which are 21 CFR compliant, which is very well accepted by all the regulators. Now there came an additional feature as far as your instrumentation is concerned. If you see, between the applicator and the photo documentation system, there came a development chamber. This development chamber has got a unique facility and a very uniqueness as far as your Article 1064 of your USP 2017 is concerned, that is for reproducibility of your metabolites at fixed RF values. Not only at the fixed RF values in your formulation, but they went one step ahead and they have introduced system suitability test. Briefly, I'll tell you what is that system suitability test, because if you are a formulator and if your formulation is rich in flavonoids, then they will give you a mixture of 2 or 3 flavonoids which you should apply on track number 1 and develop with your formulation and your passing batch and see the position of those standards which are provided. If they are at the fixed RF values, then only your regulators are going to accept your material, because they know quantification is absolutely difficult. I'll show you. Even there are WHO guidelines. WHO guidelines are also very distinct as far as your material of herbal compounds are concerned. They have also not put any stress on quantification, but they want the authentic material which can be obtained by proper documentation. For this, they have introduced a development chamber which is software driven, where you can control relative humidity. Most of the researchers in Ayurveda or for analytical work, they miss out. Okay, for most important factor today for good analytical development as far as TLC is concerned, to produce reproducible RF values, you have to control the humidity. Very important tagline. In TLC or HPTLC, the important factor is to control humidity. To control humidity, this instrument has been introduced. And this instrument works on isocratic system. Even the guidelines are very simple. The sample preparation, application, development, imaging, scanning, and what I want to show, these are the different chapters which have been introduced. And one more thing, the planar chromatography allows you to apply as many as 15 samples on one plate. That is an advantage over HPLC, because in HPLC you are doing sequential injections, which is time consuming, but here in one development itself you can do the documentation and development and complete your work using minimal solvents, and the cost of the plate is hardly 40 to 50 rupees. You are saving lot of cost in that also. And one important aspect what I want to show, this is a visible chromatography. You can take the images, and very important is your derivatization aspect of your analysis. Derivatization, that is using a derivatizing solutions which are available for different class of compounds to authenticate your material which is present in complex matrix. I want to now just highlight one other add-on technique which has been introduced in instrumentation, that is HPTLC MS interface. Many chromatographers today are interested in this. The metabolite of your interest can be put on this interface. It can be removed from this stationary phase with this device and directly injected into your MS for further identification and characterization. This has been of great importance to many researchers. Many institutes have gone for this instrument, and this is of vital importance. At the end, there is a little bit of time constraint. I want to make a small presentation on global atlas. Very important now. This global atlas we are bringing out for all our herbs which are used in herbal medicines. There are different stakeholders who are working on this global atlas. One is USP India chapter, IPC Indian Pharmacopoeial Commission India chapter, AOAC Organization India chapter, and HPTLC Association of Switzerland. They want to bring about the global atlas of your herbal medicines so that intercontinental differences in your global profiling can be very well resolved. You do not, when you buy a herbal sample or herbal raw material from Australia or US, you will find the profile to be different. And then you will have slightly element of doubt in your mind whether it is genuine or whether it is spurious. Therefore, you can go to the herbal atlas and you can verify this. This herbal atlas will bring about the unification from all the countries. All are contributing. And finally, the compilation will be done by HPTLC Association of Switzerland. With that much of insight, I wish to end my presentation here. I think I have triggered you to some extent about this very important analytical tool for your herbal work. Before I end, I wish to highlight very important point. NCROM, who are the technocrats working with CAMAC for HPTLC promotion, proper TLC promotion, have got a state-of-art lab in Mumbai, Mulund. You chromatographers, for your work, I have received many queries for that, are most welcome to our lab, where we do proper method development for you, proper method validation for you, and you can get an opportunity to work on the latest CAMAC instrument. The latest instrument about this, I have the presentation also, but since there is no time, I want to bring about. Last week, CAMAC has introduced in Asia, it was an Asia launch from our lab in Mulund, the HPTLC Pro. What is that HPTLC Pro? You saw the sequence now, application, then the development, the documentation, and the scanning. Those are different cubicles. Just like your HPLC, one instrument they have brought about with a conveyor, which will start, you have to just program it, put your proper mobile phases for development, proper command to the instrument, and all your work will be done in automation, in sequence, even up to MS level. MS level meaning the interest, the metabolite of interest at a fixed RF value, when you give a proper command, will be picked up by the interface and injected into MS. But CAMAC does not provide MS. CAMAC is not in the business of MS. This system is totally works with all the MSs available. So if you have Agilent, Waters, or whatever MS, Shimadzu MS, it is compliant with that. With this, I end my brief presentation. Thank you very much for allowing me to highlight this unique technique used for herbal work. Thank you once again.
Thank you. you, sir, for brief information about TLC and HPTLC. I request YK Gupta, sir, to felicitate Mr. Bharucha.
During discussions, if you have any questions, I'll be there to help you out. If I can help you extempore, well and good, or otherwise I'll go, which I think you can. Yes. Hopefully. Thank you very much.
So the next speaker of the session is Professor S P Joshi, ma'am. Ma'am was a scientist F in National Chemical Laboratory. She is PhD Organic Chemical from NCL. She currently serves as a consultant and scientific advisor to various pharma industries. Although she is organic chemist, but she has done enormous work in natural product drug discovery and phytochemistry. She has expertise on HPLC and other hyphenated techniques for more than 36 years. I request ma'am to start her presentation, focusing our integrative audience.
Thank you very much. I don't take much time in introduction. I will start to the subject because we have very small time. So my talk is on hyphenated techniques in chemical analysis, particularly LCMS, LCHRMS, LCMSMS, and LCNMR. Okay, what is hyphenated technique? It is combining two separate techniques with assistance of proper interface. Mostly chromatographic techniques are coupled with spectroscopic techniques. The hyphenation of these strategies allows better examination of the product and its segments. That is part of it also. It is like looking at your product from two different dimensions. At one side we see how many compounds are there, and other side we try to analyze them by some specific spectroscopic. So hyphenation method increases, that is why hyphenation method increases specificity and sensitivity of our analytical method. This term hyphenation was used, first time introduced in 1980, although it was used earlier also like in GCMS. Here it refers to online combination of separation technique and one or more spectroscopic detection technique. Like chromatographic technique produces pure or nearly pure, or maybe 80%, 90% pure fractions of chemical components in a mixture. Spectroscopy produces selective information for identification by studying the spectra or by using standards of library of spectra. The direct coupling of chromatographic techniques with spectroscopic examination of separated fraction constitutes several powerful analytical techniques, although we are going to talk about only 4 So this method helps structure elucidation of complex compound also. Now there are single hyphenated techniques where only one single chromatography technique is coupled with one single spectroscopic technique, like LCMS or LCHRMS or LCNMR. But there are also double hybrid hyphenation techniques where more than one chromatography technique is coupled with one or more than one spectroscopic technique, like LCPDA-MS, photodiode detection, then LCMS-MS, double MS, or LCNMR-MS. So this way, online coupling, we have online coupling of solid phase extraction also, where it helps hyphenation of our system, then solid phase micro extraction, or large volume injection, where even 5 gram or 10 gram of extract we can directly separate, where even small amount of minor compounds also we can identify, like solid phase extraction LCMS or LC solid phase extraction LC, popularly known as SEBOX. Although these are very important and very useful techniques, we don't have time to go through them. So let us stick to advantages of hyphenated techniques. They are fast, they give accurate analysis, we have high degree of automation, high sample throughput, very fast, then better reproducibility, then reduction in contamination due to closed system, and separation and identification is achieved same time. Separation requires a number of chromatographies, combination of column chromatography, TLC, HPTLC, preparative TLC, preparative HPLC. So all those things are avoided in this. So these are list of hyphenated techniques which are used, but as I said, we will restrict to four only, which are used in chemistry of natural products, particularly chemistry of plants, phytochemistry. So important techniques are LCMS, LCHRMS, LCMS-MS, and LCNMR. Then GCMS is also one hyphenated technique which was used from 1960 onwards, but it is only useful for small molecules, volatile molecules, and heat stable molecules. But anyway, we are not going to discuss it. So now LCMS, liquid chromatography mass spectrometry hyphenation technique. It has two components, one is LC, one is MS. Now LC is liquid chromatography separation. That is mobile phase is liquid and stationary phase is solid. But we are not going to, we are going to restrict this instrument hyphenation restrict to special liquid chromatography, which is called as high performance liquid chromatography. We are all aware of that is HPLC. Here separation is achieved at high pressure, which gives very good separation of complex mixture also. If you see history of HPLC, it was developed to separate polar molecules, including water soluble molecules, by using synthetically prepared non-polar material, which is called as reverse phase material. Then smaller particle size than whatever was traditionally used for separation. Then separation is carried out at high pressure, which gives very good separation with very good theoretical plates. This HPLC is considered ideal for separation of polar compounds such as aqueous compounds, which are particularly related to traditional medicines like decoction, tinctures. Mostly they are very polar compounds, and in that case we have to go to HPLC only. So HPLC is considered as a choice of separation method because separation is carried out at room temperature. That is why it is compatible for heat stable compounds. So heat decomposition due to heat is not there. Compatible with polar compounds, although afterwards stationary phases compatible for non-polar compounds also were developed. Then we can monitor different parameters of these things like stationary phase, column length, particle size, flow, mobile phase, and different detectors to achieve separation of almost all components from organic mixture or even sometimes inorganic mixtures also. Now second component of this hyphenated techniques is mass spectroscopy. It allows analysis of molecular weight of a compound. So one side you separate by LC and second side you identified its molecular mass. And mass is very sensitive and has wide application in different fields of organic chemistry. So in LCMS there are three components. One is liquid chromatography, that is HPLC. using a detector such as it identifies as many, almost all compounds like PDA. Then one interface to volatilize compound, to evaporate compounds, solvent from LC, and compounds are transferred to MS unit, and MS unit where various ionization techniques are used to get ionized compound, and which are analyzed by mass analyzer. So these are, as I said, different compounds of mass: ion source, analyzer, and detector system. Okay. Then there are different ionization method like electron impact, which is compatible for GCMS, fast fast atom bombardment, which is not used in hyphenation method. Then atomic pressure chemical ionization and electron spin ionization, which are compatible with hyphenation, LCMS hyphenation, and used in this, and MALDI also. Okay. So this way we can combine LC and MS, and you get one side LC and of a complex mixture, and second side mass of each and every component. Then we have high resolution mass spectroscopy also. We can couple that with LC also, where detection of analytes to nearest or 0.001%, that is 4 digit, is possible. This helps identification of exact molecular formula, not molecular formula. You can actually identify molecular formula, and hence to identify unknown compound and to elucidate structure of a molecule. Then MSMS, I think Dada Patil will talk about it. It is a two-step mass, that is mass of mass. So first, it is multiple step of mass spectroscopy. Here ions are formed in ion source and separated by mass to charge ratio in first stage of mass spectroscopy, that is MS1. Ions of then from this few ions are selected, which have good ionization capacity, and they are fragmented again, and resulting ions are then separated and detected in second stage of mass spectroscopy, that is MS2. Here mass of many compound, 2 or 3 compounds may have same splitting pattern, but MSMS is not same. It is particular, that is a fingerprint of a compound. So if you are not able to identify your compound by LCMS, you have to go one stage ahead and go to LCMSMS. Then if you have a method, then it has to go through method validation, 8 parameters like accuracy, linearity, range. It is same applicable to HPLC also. Then precision, robustness, all 8 parameters. Okay, now here I have given one small example. So I have a mixture, this I have taken from it. I have a complex mixture. So I run LC of that using some standard mobile phase, okay, and RP18 column. Then I get some 12 peaks I have got here. Now what I do, so I know my compound has 12, my mixture has 12 compounds. So that is not enough. I want to know what are those 12 compounds. So I go to LCMS. So then when I do LCMS, each mass of each 12 compounds is detected. So when I know mass of these 2 compounds, I can find out probable molecular formula of these 12 compounds. But this doesn't give me exact mass. So maybe each and every compound will give me 8 to 10 possible molecular formulae. So this is not again enough for me. I want exact molecular formula. So so here I go to LCHRMS. When I go to LCHRMS, here exact molecular formula up to 4 digits atomic mass is calculated. How it is done? Here carbon we consider 12. but hydrogen is not 1, but it is 1.007825. Oxygen is not 16, but 15.994915, so on. So I calculate possible molecular weight of particular formula. Actually, these are available on that table is available, and I compare that with my actual HRMS. So from comparing these two, I will know exact molecular formula of all my 12 compounds. I will take 2 minutes. Okay. So gas chromatography, as I said, we don't. I come to LC-NMR. LC-NMR is coming up as a new analytical tool. Here LC is attached to a NMR tube through a solid phase extraction, where solvent is evaporated, each component is passed through, solvent is evaporated, and that is washed with deuterated solvent, and that compound is in a deuterated solvent is passed through NMR probe or NMR tube. So in LC-NMR you can do 2D experiments, then carbon proton experiments, nitrogen, phosphorus, so all types of studies you can do. So by even from a mixture you can get actual NMR of each and every compound, although this system is not very sensitive, but you get actual spectra for each and every compound. NMR spectra, that is carbon spectra, proton spectra, correlated spectra. Then it is possible to have a double hybrid hyphenation technique with this also. That is that you can attach a mass via a splitter at outlet of LC-NMR interface. So same time you can get separate your mixture, you can identify that by NMR, and you can have mass or HRMS of this, and have complete structure elucidation of your compound in single experiment. So I call this as the, by using these hyphenated techniques, it is possible to identify your compounds from a complex mixture. Although you cannot use this as an analytical tool, you can take help of this and develop your analytical tool. Okay, thank you very much.
Thank you for in brief explanation. It's really hard to summarize such a vast topic in 10 to 15 minutes. I request,
really tough.
yes, it's really tough, but I'm sure that all the audience must have understood the potential of these techniques. I request YK Gupta sir to felicitate ma'am. Now the next speaker of the session is Dr. Hemant Agarwal. I would like to introduce him as an activist rather than only scientist. He is a PhD in flow cytometry, immunology from University Hospital Essen, Germany. And he is currently the director of Flow Cytometry Solutions Private Limited, where he worked as a scientist plus activist plus consultant to the various industries and explained the potential of flow cytometry. And he is most acting driving force behind Indo-US Flow Cytometry Association. I request Hemant Agarwal sir to explain the potential of flow cytometry for AYUSH research. Please,
Thank you.
sir.
Am I audible to everyone? Okay, good. So good afternoon everyone, and thanks to the organizers for inviting me to talk about flow cytometry. Just a small question, anyone have heard about flow cytometry before in this room? Okay, so we have few people, very nice. So I'll try my level best in this, have given me 15 to 20 minutes to compile the things. It's a vast branch, but I'll try my level best to give you an idea that what all you can do using flow cytometry. As this conference is all about Ayurveda and AYUSH and herbal products, so the speakers before me talked about how you can get this kind of extracts that you can utilize over the cells and check their properties like anti-cancerous properties or other properties, those like if you are interested in. So how this technology, flow cytometry, can help you to know the results that how your compound or the product is working. So those who are new to flow cytometry, this is just an introductory slide which says flow, as the name suggests, motion, cyto, cell, metry is the measurement. So we are measuring the properties of cell while they are flowing. The good thing here is that we are doing all this with a rapid pace, and it's a high throughput technology. That means you can analyze millions of cells in no time. The only prerequisite is that the cells should move one by one. And there are like inside the machine, there are lasers are there, then the detectors are there. They detect your cells and give you the idea about that how your compounds are affecting the cells. You can do functional studies, you can do phenotypical studies using this technology. Another thing, this technology is not an assay, but it is a tool which you can get the information and can do something else in the same lines. So why this cytometry mania is there? If you see, if you just type in flow cytometry word in PubMed, you can see thousands of papers open up. In today's world, everyone wants to get associated with this technology. So the reason is that I always say it's like a salt and pepper, which goes good with every cuisine of the world. So you look at any science, you just have to think about how you can utilize flow cytometry to get the results. So as you can see, it's just getting perpendicular and perpendicular the more we advance into the years. So if you see here, these are the pictures of different flow cytometers. But what makes them different is the capacity or the capability of these instruments. But what is inside this instrument is that the cells are going one by one. There's a laser which strikes the cells, and then you can get the signals in different detectors. There's a computer and the software which filters the information. You get the graphs on your screen. And now the question is how to read those graphs. So in my next slide, I'll let you know how to read those graphs. So what all we can measure using flow cytometry? Any kind of a cell, whether it's a tissue cell, blood cell, alga, protozoa, chromosome, yeast, anything. So, but it has to be within the limit of the detection limit of the machine. The conventional machines can do something between like 0.5 to 14 micrometer. Then there are the machine which can go with the macro kind of environment, and then there's something which can go with the nano kind of a thing. So the power of this technology is the millions of cells can be analyzed in no time, which can give you a lot of results which can be stored and can be reanalyzed. So this cartoon is kind of a summary which says that you can get the cells from anywhere. You run it on the machine, you get the data, and this data can be analyzed and can be interpreted in the right ways to get the right results. So how the cells are studied with this technology? We can study using the scatter properties in the form of forward scatter, in short FSC, or the side scatter, in short the SSC. So it can give us the idea about cell complexity, and this gives the idea about the size of the cell. Then if we add something from outside that we call exogenous, that could be the form of the antibodies which are bound with the fluorescence. So lasers are there, they strike the fluorescence, you get the signal. Or it could be an autofluorescence of the cells which can give you the idea. Like for example, the classical is GFP. You can study the GFP labeled cells without any kind of a exogenous staining. And with FSC, as you can see, this is a typical blood where you can get lymphocytes, monocytes, and the neutrophils without any staining. Or you can get the fluorescence. These are the cells which are not binding my antibodies labeled with the FITC, and these are the cells which are binding my antibodies labeled with FITC. Or I can have a look in the 2D plot. I can get double negative, double positive, single positive, and the single positive. So how we can apply this technology in Ayurveda research? If you look at this, I have downloaded few papers where you can see these guys have utilized flow cytometry. But what they are mainly doing, they are utilizing apoptosis assay or a cell cycle assay. But they are not really harnessing the power of this high throughput technology. So I give you the idea what all other applications can be done using flow cytometry. So this is the list, which is not a very exhaustive list. It's just the sum of the applications out of hundreds of applications which can be done using flow cytometry. So cell phenotyping, in a mixture of cell, I can identify a different kind of cells on a single cell basis. For example, if I'm interested in this stem cell, I can analyze this cell using the antibodies against the cells. So for example, this is a flow cytometry data. In this particular thing, I'm looking for the cells which are activated or non-activated. CD25 is a marker for activation, and if the cells are negative, they are not activated, and there the cells are activated. We can look for the activation in a different form if their size or the granularity changes. So without doing any staining, I can come to know with the effect of IL-7, my cells are getting activated. Or I can have activation markers like CD69, CD71. Depending on the cell type that I'm using, I can choose the different markers. Cell cycle, as I told you, it's a very well utilized assay in Ayurveda research, where people are looking at the different phases of cell cycle. And if you have a drug which is anti-cancers, you can do a arrest. You can look at the cell arrest. For example, in this case, the cell arrest is happening at the S phase. In this case, the cell arrest is happening at the G2M phase, just by using a single dye which is against the nucleic acid like DNA. And you can also look at the cell viability by using a traditional dyes like preputium iodide, 7-AAD. So my dead cells will take the PI, and my non-dead cells where the membrane is still intact will not take the PI. And very easily I can find out these are the dead cells and these are the live cells. And in case of apoptosis, I can just have a look at the scatter plots. The ones which are having a low FSC and high SSC could be my apoptotic cells. And the ones which are having a high FSC and low SSC will be my live cells. So apoptotic assays can be done in a different ways. So that is the scatter plot, or you can use annexin 5 PI to look at the early apoptotic cells. Or you can have a sub G0, G1 peak where the DNA fragmentation is happening or the loss of DNA is there. You can think that this is peak as a apoptotic peak, and you can get the data which can be analyzed further. Or you can do active caspase 3 assays by using flow cytometry. But what is here relevant is that we are doing all the studies on a single cell basis. It is not like a mixture of cells. For example, Western blots. You will never come to know which cell was producing how much of the protein. But here you will come to know that each cell, whether how many are responders, how many are non-responders. So here another thing is the mitochondrial membrane potential we can do by using a dye like JC1. On a single plot you will come to know how many cells are having a change in MMP and how many cells are not having the change in MMP. Then you can have a look at the ROS species, which is also a very good test that can be utilized in Ayurveda research, or the cell proliferation. The traditional assays like MMT assay, where I will never come to know how many cells are proliferating. But here, if you look at this assay, I'll even come to know how many generations are producing and how many cells are there in every generation. Or if you just want to know yes or no whether cells are proliferating or not, then you can look for something like a thymidine analogs like BRDU or EDU. And you can even look at the phosphorylation of the proteins by using flow cytometry. We call it a phosphoflow. Or you can do even with, like for example, we do ELISAs for the cytokines. We need not to do ELISAs because in ELISA we don't know how many cells produce the cytokine. But here if we know intracellular staining, I'll come to know which cells were producing, let's say in this case IL-12, and how many cells were producing TNF alpha on per cell basis. And if you want to look at the many different analytes in a very small amount of supernatrins, then there's a technology known as multiplex bead arrays, which can also be integrated with flow cytometry. In a single go, you can have a look at around 80 analytes, that too in just like 10-20 microliters of a sample. Then there are sorting application. You can sort the cells and you can do whatever you want with the sorted cells. You can have genomics, transcriptomics, metabolomics, proteomics, or you can do anything you want with the sorted cell. So this is the only technology which can take out the cell from a mixture and you can do the things that you want as per your research idea. Then this is the new thing which in flow cytometry we call it image flow cytometry. Rather than just getting the dots on the plot, if I can get the image also, that can change my life. So this is, we call it image flow cytometry, and in the image flow cytometry we can have something like co-localization, cell-cell interaction, cell cycle and mitosis. All the images can be produced. Though these images are not very high resolution, but it will give you the idea where your antigen is lying, whether it's inside the cell, outside the cell, what is really happening. So as you know that by this time that it has lot of capability. But as Uncle Ben said in Spider-Man, with great power comes the great responsibilities. And that's the Peter Parker's principle. And yes, with this kind of a technology, we have to be very, very careful, because at any stage, if we misinterpret our data, the things will entirely change. So how? So there was a Amgen company in America which took the 53 landmark papers in cancer biology and tried to reproduce it. But you know how many papers they reproduced? Only 6 papers. There's another one where they took the academic research and they reproduced only 25% of it. So what it does mean to us if we want to utilize this technology as a tool in our research. So if you see this bar graphs, here you can see the analysis, sample preparation, operator, setup, reagents, acquisition. There are multiple factors which play a role, and analysis is a very, very big factor. How we are analyzing our data. Though we have the very $1 million machine in our department, but if I really don't know how to really analyze the data, how to do the good sample preparation, I really cannot do anything, at least in this field. So if you see, this is some examples of poor data analysis and presentation, and this is the data which is already published in very good journals. It is not that these are not published data. Here you can clearly see, though they are trying to show apoptosis assay, but I really cannot see, just like a kind of a debris are everywhere. I really cannot make it out whether there's any apoptosis happening. Similarly, here they want to know how many dead cells are there, but the kind of a gating they are doing is incorrect. And here they want to know the cell cycle assay, but this is a control. The condition of a control itself is not appropriate to be considered as a control for any kind of a drug treatments or analysis. So this is just a few examples out of thousands of examples where people publish flow cytometry data, but they really don't know what they have published. So in the let me come to this slide, then I will let you know a few things that how we can be successful and do correct flow cytometry. We have to know how to design the experiment correctly, how to run it correctly, and how to analyze it correctly by following these kind of things. So to do good flow cytometry, this is my kind of a tagline, and not do flaw cytometry. It's very easy to change your flow into flaw. So summary is, the flow cytometry is a quickly expanding technology and is powerful tool in modern biological research, has numerous applications and can contribute immensely in Ayurveda research. So my idea of understanding the flow cytometry when I was a postdoc is that if I can understand this technology in a great way, I can do wonders in my research. So with that idea in my mind, I started my organization known as Flow Cytometry Solutions, which help people in understanding this technology in a correct way. We have thousands of installations all over India. But if you say that how many people really know this technology, I can say only a handful of people. Only a handful of people. There are many, many examples which comes in front of me every day where just people are wasting the money and the time utilizing technology without having the proper knowledge. So with these words, I want to say that we do a lot of promotion of this flow cytometry technology, and in the conjunction of the same thing, in the month of January and February, we are doing a lot of workshops at four different areas all over India to teach the young people and the people, those who are already in this field, different aspects of flow cytometry. So I invite all of you to come to this workshop and see the kind of a people we brought in from US or India, and they try to give as much as possible so that you can improve your research over it. So with these words, I would say thank you so much for inviting me again. Thank you. Any questions you have, I would be very happy to answer if there's any time.
We can take
Okay, no problem. Thank you so much.
Thank you so much, sir, for covering the important topics about facts. I request YK Gupta sir to felicitate Hemant Agarwal sir.
Thank you.
Where is it from? This
Jaipur. Yeah, I'm based.
I think your statement was really,
Thank you. Thank you so much.
70% is not doing properly.
Exactly. Thanks.